to show you a research tour from CUSABIO. Here's the CUSABIO website. In the technical research part, we have research tours. In the research tour page, we have different types of research tours that can be convenient for your research. But today, I will introduce this one, the endonucleoside analysis tour. So it basically is a restriction site finder tour. So you can quickly enter a nucleic acid sequence, and this tour will help you to find and count restriction sites. So the result will be shown with enzyme name. So let me quickly show you an example. I have a DNA sequence that I saved here. Let me paste the DNA sequence in this box. Okay, so right now we have the sequence here, and we click the find restriction sites. So as you see in this summary table, we have 4 different enzymes that can cut this DNA sequence, and the recognition sequence for each enzyme are listed here. We also have the cut site list, and even we have the count number. So for example, for this enzyme, there are 2 cut sites in this DNA sequence, and also the position are shown in the table. So for each cut site. And if you want to know more, we have a very detailed instruction here. But basically, just enter sequence, click the find restriction site button, and then view the result. We also offer another list of the supported restriction sites. So this is the enzyme list. So as you see, we have so many enzymes, and in this list here, we have the recognition sequence for each enzyme, and also we have the cut site. So if you are interested to know more, please check this checklist page and take a look. So in summary, this restriction site finder tour is a very convenient tool to help you to find the cut enzyme, and also to find the cut site and know what the consequent count. Yeah, hopefully you enjoy it.